So I'd like to talk to you today about use of human FcRn transgenic mice and how it will aid in the selection of antibody therapeutics and predict their clearance in humans. And amongst the stages of antibody development, it starts off with target validation and molecule selection, whether it's binding, determining the affinity and selecting the binding to the target, as well as affinity for human FcRn binding. These different assays are available, sequence-based, 3D structure, flow cytometry, surface plasmon resonance, and so on. And once the set of antibody candidates has been reduced down for those with the best characteristics for targeting the molecule of interest, lead optimization involves the characterization of PK. And this requires using biological systems and using mouse disease models if you're going to do efficacy studies. And if you're going to go forward into clinical development, you'll want to test in human FcRn transgenic models or the non-human primate. And what we can offer are models that use the human FcRn transgenic mice. And these models here are what we can offer. And up the top left is the TG32 mouse, and it lacks mouse FcRn, and it has a human FcRn promoter that's driving the expression, and it yields half-life that's closest to the human values. And I'll show some data how well it correlates to non-human primate and human data. Another version of this mouse, the TG32 SCID mouse, has the SCID mutation in the PRKDC gene so that it's immunocompromised. And this model is utilized if you want to avoid anti-drug antibodies. The next model is the TG32 HFC mouse. And what it has is it's transgenic for the human IgG domain that it allows it to express human IgG1. And compared to the TG32 mouse, which only has mouse IgG antibodies, they have no affinity for human FcRn. So there's no competition within that biological system compared to the TG32 HFC mouse. And that competition can be of interest to know how well your therapeutic will perform in, again, a more humanized system where competition exists. And I'll share some data in a few slides. The next mouse is the TG32 mouse, which is deficient for mouse albumin. And the utility of this mouse is that mouse albumin has a high affinity for human FcRn. And if mouse albumin is present, if you do PK studies with albumin therapeutics, the human albumin, your candidates, they can't compete with the mouse albumin. So they're artificially accelerated in their clearance. And so by removing mouse albumin from the system, you then get a better estimate of the human PK that your therapeutic would yield if it's albumin-based. And again, I'll show data for that as well. And then there's the NSG TG32 mouse. And NSG mice provide human FcRn on the NSG background for human cell engraftment. And we'll see some of this presented in the next talk in terms of the use of the NSG model. And I'll share some PK data using this mouse as well. And the last model is the TG276. And this mouse, it has ubiquitous expression using the CAG promoter. And the outcome is that it has shorter half-life value that correlate well with human. And the outcome of this is that this model does a great job of discriminating subtle PK differences. And I'll show you an example of that in the next slide. So in the upper left, we did a study in TG32 mice, 3 mice per treatment group. And these are different variants of rituximab. And you can see rituximab at the top. And this is a plasma concentration time curve. And you can see that each of the variants falling down below with different levels of clearance. To the right of that is some data, to a pair of graphs here that shows 2 variants. The closed symbol is the variant 1, and the open symbols are the variant 2. And we have homozygous data on the left and hemizygous data on the right. And of these 2 variants, you can see when we put them into homozygous TG32 mice, it doesn't discriminate that well. And when you switch over to the hemizygous version of the TG32, the same occurs. The discrimination is limited in terms of the plasma concentration time curves. However, when you go into the less efficient FcRn function that exists in the TG276 mouse, and yet it's still human FcRn that's binding, so it correlates well with human data, you can see that variant 1 and variant 2 data separate. And the same is true when you switch over to the hemizygous version of the TG276. So that's the utility of using the TG276 if you have multiple variants and their data may be really well matched. You might want to consider using this mouse for characterizing the PK. I want an analogy of this is that if you had, instead of 2 antibodies, if you had 2 runners and they were well matched and you put them in a race and they'd have to go a great distance in order to see a difference. And so maybe if this study went out far enough, these 2 would separate. However, using the TG276 is analogous to having the runners run up a steep incline in their race. And in that way, in a shorter time, you get them to separate and show their differences. In this case, of course, PK differences. To the right of this is the NSG TG32 data. In the NSG mouse, there's an Fc gamma receptor that's a unique allele. So it binds human Ig to high affinity. And what that does is it's an antibody sink, but it has limited capacity. So what we did was we preloaded mice with human IgG, and that binds up those receptors, and the excess antibody then can be protected by the human FcRn in these mice. And so when preloaded, you get better clearance. And when you don't preload, the clearance is a little more rapid. So that's something to keep in mind with this model. In the bottom left, we've got the TG32 skid mice. And what we did was we put etanercept into both the TG32 skid as well as the TG32. And you can see that both yield the same plasma concentration curves and the same clearance rates. So this model, the TG32 skid, is a good possibility to substitute and use if you want to avoid anti-drug antibodies against your candidates. Of course, if anti-drug antibodies arise in your study and your plasma concentration time curves are abbreviated, your PK data will suffer. And so using this model is something definitely to consider. To the right of that is the TG32 HFC, and this is the model of TG32 that expresses human IgG1. This is some of our published data from a year ago. I neglected to put the reference in. We can make that available if you wish. In this study here, we put trastuzumab into TG32 naive mice, and this mouse has no competition. No antibodies are binding to the human FcRn expressed. Only the trastuzumab that is administered. And the mice that are TG32 HFC, when they're naive, they're expressing about 600 micrograms per mil human IgG1, and you can see that competition accelerates the clearance a little bit. We also immunized some of those mice so that their level of human IgG1 would go up, and at about 4 mg per mil of the human IgG1, the trastuzumab was eliminated a little bit more. And we artificially caused competition by preloading some TG32 mice with human IgG, and they were preloaded at 200 mg per kilogram. And you can see that elimination in that situation was even more. And so this demonstrates the impact of competition for being protected by human FcRn, and how the TG32 HFC mouse could be utilized if you want to see how your antibody therapeutics perform within a system that has competition involved. The last model I'm showing here is a TG32 albumin knockout, and the molecule being studied is albiglutide, which is an albumin molecule with a glucagon-like peptide 1 that's part of the construct. And in the TG32 mouse, where mouse albumin is present, it competes readily, and that causes the albiglutide to be readily eliminated. It's protected a little better than the FcRn knockout, but only somewhat. So when you then characterize this molecule in TG32 albumin deficient mice, you can see that it's well protected because there's no competition from the mouse albumin. So let's now look at the models and see how they perform against sets of antibodies. And Avery did a comparison across a large set of antibodies, and they ordered their antibodies in terms of clearance from lowest to highest clearance values. And a subset of those antibodies, they plotted data from humans. And you can see that the pattern of clearance is similar between human and non-human primate. However, when plotted against clearance values that were yielded from wild-type mice, you can see that it doesn't yield a very good pattern that matches either the human or non-human primate. They also plotted the non-human primate clearance versus TG32 home, and you can see that it correlated quite well. The same was true when it was the clearance values from non-human primate were plotted against TG32 hemizygous mice at 0.91. However, the correlation was poor against wild-type mice when they were administered these same antibodies. So the take home from that is the TG32 mouse does well to yield data that lines up well with a non-human primate, which is the gold standard for PK prediction in humans. Haraya did a similar study using these antibodies here, this set, and they compared half-lives in humans. In this case, they compared it versus non-human primate, and it yielded a good correlation of 0.75, and a similar or better correlation when compared to TG276 mice. And when the half-life of humans was compared to those yielded from wild-type mice, you can see that it yielded a rather poor correlation. So this data demonstrates that the TG276 also is a good predictor of human PK. I also want to share a PK case study that was done in collaboration with Jeff Hale from Mabsolve, and he provided a QUM FC variant. The QUM FC variant has a silenced FC. It does not bind to FC gamma receptor or C1q, and the activity is less binding than for the LALA variant. The test antibodies that we used were rituximab, either wild-type FC, the QUM, or the LALA, and we included the TG32 HFC mouse, which we immunized to boost the endogenous human IgG level. We also included the TG32 that does not express human IgG1 and wild-type mice. We boosted the mice with DMPK-LH 4 weeks before the PK study, as well as again 2 weeks before the PK study began. And the mice were dosed 5 mg per kg, and blood samples were collected as indicated. And we did ELISA analysis of human IgG, but we also did rituximab analysis using an anti-idiotype ELISA, and that's because the TG32 HFC mice have that endogenous human IgG1. So you need that anti-idiotype ELISA in order to quantify the rituximab specifically. So the human IgG ELISA, again, no data from the TG32 HFC, but TG32 yielded good plasma concentration time curves, both for the wild type as well as the QUM, and the LALA indicated that there was some ADA present because the curves showed accelerated clearance, likely due to antibody complex formation. And in black 6 mice, we see, again, good curves as well as an indication of ADA occurring in those mice as well. And you can see from the average data that the different FC variants performed very well in the TG32 mice. They line up quite well with the wild type when plotted out as a percent test article remaining compared to day 1. And here's the half-life data. You can see they're rather well grouped, and that the wild type mouse yielded even higher half-life. So it's not necessarily a good predictor. When we go to the anti-idiotype ELISA data, now we have data from the TG32 HFC mice, and you can see that the clearance is a little more rapid, and that's because of that competition from the endogenous human IgG1. And that's true for all 3 versions of the rituximab. And we see, just as with the human IgG data, we see that there's evidence of ADA for the LALA version, both in the TG32 as well as the black 6 mice. And again, when this data is also plotted on a percent test article remaining, you can see that each of the variants holds up well. And in particular, the QUM variant does well in terms of being just like the wild type rituximab. And here's the TG32 data, and it's well matched for all 3 variants. And the half-life is reduced in the TG32 HFC mouse. And again, we have wild type data that's a little more variable and not a good predictor. And when we analyze the ADA activity, we can see that the wild type antibody did not yield ADA that we could detect. The QUM yielded some ADA activity in the TG32 mouse. And the LALA variant yielded that same ADA activity in both the TG32 and the black 6 mouse. And it's of interest that the TG32 HFC mouse yielded very little or no ADA activity, likely due to it being tolerized to human IgG. And here's a summary of the PK data, and I'll just kind of summarize that as the QUM yields a half-life similar to the rituximab wild type in TG32 mice, so that that variant does not impact the PK, and the QUM competes well for FcRn protection in the TG32 HFC mouse. And that's this here. It's yielding very similar outcomes, and you can see it here as well. So I'd like to thank Jeff Hale and Ian Wilkinson for collaborating with us on this project and providing the QUM and other reagents for us to characterize. And I'd also like to thank Ilano Gonzalo-Gil, who's the study director for this project, and Daniel Villarreal, who is the project manager, as well as Caitlin, Rachel, Beth, and of course, Adriano Flora, who got the collaboration put together for us. So thank you.